Thank you. The next speaker is Dr. Adam Stewart, talking on non-antibiotic drugs. Can also lead to antibiotic resistance. And Adam is a microbiologist and researcher with David Patterson. Research interests include the effect of new and old antibiotics on gut microbiota and clinical trials on mono and combination therapy to treat CRE.
So again, thank you very much to the organisers for inviting me to come and talk today. So I'm an infectious diseases advanced trainee and doing a PhD with Professor David Patterson, and I've been asked to talk about this very interesting topic that's quite novel and is certainly going to lead to some pretty fascinating research in the future. So what we'll try and cover today really centres around 5 important points. So I'll just explain briefly about how the plot thickens in terms of the public health problem, the global burden of antimicrobial resistance, and this might extend to non-antimicrobial medications. And then I'll just briefly talk about silencing the noise and the difficulties in identifying some of these drug-disease links. We'll go through the likely culprits, everything from PPIs to beta blockers, and then making sense of it all. Doesn't really matter, putting it into a clinical and global perspective, and then some take-home messages and where to go to from here. So the understanding of the human microbiome has increased dramatically. As Sam Marie pointed out, we now have a good understanding of the microbiome composition across different body locations, both in a particular patient or person and sort of into individual variability. And this has largely been fueled by advances in metagenomics and associated data analysis pipelines. And so now we're seeing more associations between sort of microbiome shifts and lifestyle, diet and disease. So as we're all aware, emergence and spread of antimicrobial resistance is largely due to the use, misuse and overuse of antimicrobials in the clinical, vet and agricultural settings. Exposure of microorganisms to sub-inhibitory concentrations of these antimicrobials is a key driver of antibiotic resistance and is a problem. But what we need to understand is that our human microbiome is exposed to both antibiotic and non-antibiotic drugs, and how this sort of affects our gut flora and our human microbiome. So recently, non-antibiotic drugs has emerged as a potential key contributor determining microbiota composition. Our current knowledge of their effect on our gut commensals is scarce. The role of non-antibiotics drugs in the emergence of the resistance thus has received very little attention in the literature. So the plot certainly does thicken, and certainly non-antibiotic microbials, which sort of, as far as general pharmaceutical consumption, make up over 95% of intake, it could be a key driver of antimicrobial resistance globally. So there's been a few key signals that should have pointed us in this direction. So we know that polypharmacy increases the rates of gastrointestinal disorders, and things can mimic, disease states like irritable bowel syndrome. Known human-targeted drugs with anti-commensal activity have been known to have antibiotic-like side effects in humans. Then there's some old drugs, like the first marketed antidepressant, which was a monoamine oxidase inhibitor called iproniazid, was known to have a sort of antimycobacterial effect and was repurposed as a tuberculostatic antibiotic. And then the antipsychotics, the largest class being the phenothiazines, first were identified for their anti-infective properties prior to be sort of repurposed as an antipsychotic. And then selective serotonin reuptake inhibitors are known in vitro to inhibit bacterial efflux pumps. More drug classes, so orafenib, which was a drug used to treat rheumatoid arthritis, is known to have broad spectrum bactericidal activity. Clomiphene, which is an inducer of ovulation, is known to inhibit conserved bacterial enzymes involved in synthesis of bacterial cell wall precursors. So this leads us sort of to the generalised assumption that human targeted medicines are bound to have an effect on our microbiome, but it leads us to a number of questions. Are these effects direct? And if they are, what is the precise bacterial target of the drug? And is the drug effect on microbes part of its primary mode of action, or is it just an undesired side effect? So a number of drug classes have been sort of probed and associated with microbiome shifts, things like proton pump inhibitors, non-steroidal anti-inflammatories and so forth. But really it sort of begs the question and sort of indicates the ramification both for human health and for drug development, given the fact that we're facing increasing sort of drug consumption worldwide. So these drugs are also recognised as emerging contaminants and are ubiquitously detected in various environments, including wastewater and even our drinking water, sort of even looking at it from a public health point of view once again. So now I just want to talk a bit about the difficulties in sort of these studies in this area of research and how we can potentially silence the noise. So it is an important area of research, but there's a lot of confounders and unknowns that are present. So it's often unclear whether the reported microbiome effects are part of a primary mechanism of action or side effect. And a perfect example of this is looking at some psychotropic drugs which induce weight gain and then subsequent sort of shifts in the microbiome, and whether that's due to the weight gain or the drug itself needs to be elucidated. So drugs in themselves, or most of the drugs, are not known to target the gut directly, apart from some IBD drugs and drugs to treat colorectal cancer and so on. So therefore, their concentrations in the small intestine or colon are largely unknown, particularly oral medication. We deduce that sort of the intraluminal concentrations are expected to be much higher, and this is especially important for drugs that undergo biliary excretion in the colon, where there's substantial sort of water reabsorption. The exposure time is likely to be prolonged as well. And then I've talked about the local water retention and fluid shifts. So it makes it very, very difficult to interpret in vitro studies. So further complicating, defining a link, often only a limited amount of sort of representative bacterial species are assessed. And then looking at clinical sort of in vivo settings, often, changes are demonstrated in patients who have already received antimicrobials. And I think the best example of this is in our sort of hematology and oncology patients, particularly stem cell transplant patients. And often these patients as well are receiving combinations of these non-antibiotic drugs, further complicating things. So all of this points to us to sort of create more rigorous studies sort of to account for these confounders. So things like, with example, looking at metformin, using fecal transfer from metformin exposed individuals into germ-free mice, and then demonstrating improvement of glucose tolerance, and as well as observing this when it's introduced into the colon and thereby eliminating metabolism. So you can see it starts to become quite complex to remove some of these confounders to sort of demonstrate a direct effect on the human, sort of the gut microbiome. So these non-antibiotic drugs really have alter our microbiome right right through our gastrointestinal system. So pH changes for PPIs, cytotoxicity from non-steroidals and PPIs, changes in gut blood flow, interactions with sort of various transporters and so on. So there's a whole host of things that we've got to sort of take into account. So there is limited sort of data in this space, and it is evolving. But here is just a number of different sort of non-antibiotic drug compounds that have been tested both in the human and the animal setting. And as you can sort of see, it's the low-hanging fruit of the PPIs and the metformins and non-steroidals, corticosteroids that sort of get targeted early. So I just want to have a look at this key publication in Nature last year that's probably the best surrounding this sort of sphere of research. They looked at 40 representative isolates and then screened them with just over 1,100 sort of compounds in a sort of generated anaerobic medium broth. And this was meant to partially recapitulate species in relative abundances representing the human gut microbiome. And they showed that 24% of the non-antibiotic compounds, so these human-targeted drugs, were active. And what they defined as active was at least one hit, so inhibiting growth of at least one bacterial species. Most were effective only against a few strains. 40 drugs affected at least 10 strains, 14 which have not been previously reported to have direct antibacterial activity. So these are incredibly important for drugs not on that previous list that I showed you, to gain knowledge of their effect on our microbiome. So there was a bacterial species sort of differential response that was observed. So abundant sort of Roseburia, Eubacterium and Bacteroides were the most sensitive, and the gamma proteobacteria being the most resistant to these drugs. Very similar to what we see with antimicrobial effects on the gut microbiome. So species with higher relative abundance across healthy individuals were significantly more susceptible to these human-targeted drugs. So then they explored the question about sort of physiological concentrations. So they screened everything with 20 micromoles of a sort of standard concentration, but then they hoped to sort of model this against what you'd expect to have intraluminally. And they deduced colon concentrations based on drug excretion patterns from previous published work. And essentially they showed that at these higher concentrations, and particularly drugs that have lower plasma concentrations and thus higher intraluminal concentrations, they certainly had a higher sort of hit rate, as it were, on the bacteria. So just for example, metformin reaches 10 to 40 micromoles in the plasma, but its small intestinal concentration is 30 to 300-fold, thus increasing its anti-commensal activity. So therefore, screening studies are likely to grossly underreport impact of non-antibiotic drugs. So then they took a step back and looked at some clinical data and then looked at the human drugs with anti-commensal activity against sort of antibiotic-like side effects in humans, and found those with anti-commensal activity were more likely to have antibiotic-like side effects in sort of placebo-controlled trials. So then narrowing it further, really the majority of these drugs seem to sort of align with the anti-metabolites, antipsychotics and calcium channel blockers, only some of which a clear mechanism may be relevant. Anti-metabolites incorporate into the RNA or DNA, interact with these synthesis enzymes, and then become cytotoxic. So you can see that if there was some conservation of some of these drug targets with bacteria, it's easy to see how they'd become sort of bactericidal. But then looking at some of these antipsychotics, there's never been dopamine or serotonin receptors identified within bacteria, which sort of leads to an off-target sort of mechanism. So now on to the issue of resistance. So there's a strong correlation between resistance to the antibacterial agents and to these human-targeted drugs that can't simply be explained by general cell envelope composition. So it spans both gram-positive and gram-negative organisms. So they looked at E. coli and this Tol-C sort of multi-drug efflux pump resistance gene and compared it to a wild type, and they put it through a chemical library rather, and they found that the E. coli lacking this resistance gene was not only more sensitive to antibacterials, but also equally more sensitive to these human-targeted drugs. So there's certainly a link or a signal of a link there. But this doesn't explain everything. So in in bacteria like Clostridium and P. distantinosus, strong antibiotic resistance contrasted relatively weaker with these human-targeted drugs. So it leads us to step back and say that there's an important distinction between specific antibiotic resistance mechanisms and then these generalised resistance mechanisms that may be conserved across antibiotic and non-antibiotic drugs. And this is a bit of a complex diagram, but I'd like you to focus on section A. You can see on the x-axis the number of antibacterial drugs with anti-commensal activity, and on the y, the human-targeted drugs. And you can see that E. coli, with loss of that Tol-C efflux pump, it sort of follows that regression line down, being more susceptible both to antibiotic and non-antibiotic drugs. With a Spearman correlation coefficient of 0.6. So looking at mechanisms now, they screened a genome-wide overexpression library in E. coli against seven non-antibiotics, and they showed that overexpression of this Tol-C rescued those bacteria and enabled growth across all tested drugs. So there does seem to be an overlap of resistance mechanisms. So moving on just quickly to cancer chemotherapeutic agents, we know that this is important, an important patient group, and that the changes in the gut microbiome contribute to development of sepsis. We also know it's been well studied about the microbiome changes in hematopoietic stem cell transplant patients, and some of those sort of domination of specific species of bacteria. So I just want to focus quickly on this SOS response that's triggered by DNA damage that may promote cross resistance between both non-antibiotic and antibiotic drugs, with increased spread of antibiotic resistance genes, particularly among fluoroquinolone and rifamycin classes of antibiotic. So this is just really explaining how ciprofloxacin works, inducing an SOS response, which is sort of a conserved pathway with some of these, or a conserved response with some of these chemotherapeutic agents. So the rate of horizontal gene transfer is certainly increased during this SOS response. And then just quickly, antibiotics frequently used for prophylaxis in the cancer patient population are also the strongest activators of this response. And then platinum, not much has been done in platinum, but there is a correlation between sort of heavy metal resistance genes and antibiotic resistance genes as well. And then integrating it all together, you start off with a healthy gut microbiome. There's mucositis, dysbiosis, induction of this SOS mediated mutagenesis, with the addition of antibiotics selecting for resistant bacteria, which leads to translocation and bacteremia and sepsis. And then quickly, just a UQ group that deserves a plug. They looked at non-antibiotic drugs inducing resistance by direct conjugation and exchange of genetic material. They looked at a number of drugs, but essentially used a donor E. coli with a donor RP4 plasmid and a Pseudomonas putida, mixed them, added the pharmaceutical, and then looked at evidence of conjugation and resistance, and found that there indeed was enhanced sharing of genes. Proton pump inhibitors are well known to alter the microbiome, and certainly there was a recent study that showed higher rates of ESBL rectal carriage in hospital admission. Similarly with metformin, but once again, very difficult to tease out obesity, type 2 diabetes effects on the microbiome. So wrapping up, does it all matter? So there are a number of reasons for concern. So these drugs are over 95% of what we take globally, and consumption is large and likely to increase. So, and although evidence is still forming, there's general acceptance that non-antibiotic drugs can change microbial diversity, and if anything, is likely to be an underestimate. There is preliminary data indicating a role of development of resistance. Even if it is a small role, it's all going to be compounded by mass drug consumption. So they're therefore very important to describe. So all of these drugs that have been reported to have some effect on resistance also have an incredibly positive public health benefit in terms of mortality. But I think the lowest hanging fruit is the PPIs, which can potentially come off. So then there's been interest in these new pipelines of drug development working in parallel with microbiome association studies to really determine their broader effect. And then this all leads to potential translational applications in terms of controlling antibiotic resistance, improving drug efficacy, and potentially mitigating drug side effects. So it is more than just antimicrobials. It's an important area of growing research, but very difficult to tease out causality. We need to consider microbiome changes and drivers of resistance in a broader context in terms of drug discovery. So thank you very much.